Hello students, and welcome back to the bio video thingy. Welcome to my backyard where invasive species vines are taking over. I have tried to get rid of them. They took over the whole fence. Today we are going to be talking about the topic of DNA replication. Replication is a fancy word for taking one thing and making a copy. Now, in DNA replication, you want to make sure that your copy is exact, because if you are just slightly off with your copying, mutations are bound to occur. In fact, DNA replication is going to be your other top source of mutations within a cell's DNA, beyond the obvious mutagens, such as UV radiation. I should go put on some sunscreen. So this process of DNA replication, or making a copy of your DNA, is going to happen in 4 distinct steps: formation, initiation, elongation, and termination. Of course, like all chemical reactions in the cell, it does not happen by itself. In fact, it happens with a whole host of enzymes, all of which end in the lovely words ACE. And for work this precise, you know what the body uses as their chemical tools. Enzymes. You thought you escaped them. You did not. Enzymes are going to speed up chemical reactions so that we lower the activation energy. They also are very specific protein shapes, so they're excellent at holding DNA in place, unwinding it, and just making sure the whole process goes very smoothly. So I have taken the liberty of drawing an analogy between all of these enzyme tools that help us out with DNA replication and some different household tools that help me out with jobs around the house. Before I show off to you exactly what's going on, let's get those notebooks set up. So in class so far, you probably have already figured out that our main topic for today is DNA replication. Of course, the main goal of DNA replication is to make an exact copy of your DNA. I'm going to call that as creating 2 identical daughter strands from 1 parent strand of DNA. I'm actually going to go ahead and divide the rest of my page up to capture some of the other thoughts that have come up in class so far. The first one being that this is a semi-conservative process of replication. What this means, and what we saw in the case of Millicent and Stahl, is that the new DNA strands are made of 1 old parent strand and 1 new daughter strand. Getting ready for the process of copying the DNA. And in order to do that, of course, you want just like 1 single template strand that you can read across and copy every nucleotide. Make sure there's a perfect match. Of course, you also have to remember that DNA is tightly wound inside of chromosomes, much like a ball of yarn. Nice, neat package that fits into your nucleus and also keeps the DNA safe and locked away when you don't need it to be expressed. So step number 1, I actually have to unwind my chromosome so that I can access my DNA and read it and make a copy. Of course, this process needs to be very guided and carefully done, otherwise you could end up something like the inside of my knitting bag. You didn't see that. So if we want to think about this, DNA is actually 2 strands, right, in our double helix. And these strands are going to wind around each other, forming a beautiful helical structure, a little phosphate sugar backbone on the outside, etc., etc. But this becomes a problem when we actually need to replicate or make a copy of the DNA. Because all of those nucleotides, there's like A, T, C's, and G's, they're all in the interior portion of the DNA there. And they can't be read when it's all twisted up. I also run into a problem because if I try to open up the DNA and read it, I start twisting it even worse as I go along. So we have an enzyme that is actually going to work as something that unwinds our DNA, allowing us to actually read it and open it up. This enzyme is called topoisomerase. Once topoisomerase has done his job, I'm going to have a very nice quote-unquote flattened piece of DNA where I have 2 strands lying next to each other and they're not twisted together. Now, if I remember from DNA structure, all of the code actually for DNA that I need to copy is on the inside. let's take a look to refresh our memories. So we had the sugar phosphate backbones on the outside strands of the DNA, and they're connected on the middle, their nitrogenous bases with the actual code, A, T, C's, and G's, by really weak hydrogen bonds. And this is where the chemical structure is actually very helpful, because in order to get onto the inside and read the A's, T's, C's, and G's, I'm actually going to need to open up the DNA strands and separate it into two separate template strands. The enzyme that helps us unzip our DNA strands and separate them into two template strands is called helicase. Helicase is the one that unzips your genes. Now, what I have here, where I have half of my parent strand still bound in the center by hydrogen bonds and half of it separated into two template strands, is also sometimes called a replication fork. A fork just means that there is a split in something, just like we can see with our DNA. I'm sorry. Was I supposed to be teaching you something? Right. DNA replication. Let's take a look at this whole zipper and replication fork idea with some actual nucleotides so it hopefully makes a little more sense. Here we go. Step 1 of DNA replication. We've unwound our DNA using topoisomerase, and now it looks kind of like this. I have one strand going from 3' to 5' with a series of nucleotides, A, T, C, and G. I have a second strand that is anti-parallel, going from 3 to 5', series of A, T, C's, and G's. And every base is connected to its complement on the other strand with a hydrogen bond, indicated by the dashed line. The first thing that we need to do is separate these two strands so we can actually read this code. To do that, I am going to use the enzyme called helicase, who's going to make a replication fork. We can also think of helicase not just as a zipper, but as a pair of scissors. Whoa, it's windy out here. And in this case, helicase is going to get to work breaking up some hydrogen bonds, separating my DNA double helix into a replication fork. Let's get all of these concepts down on page 56, the next page, as steps of DNA replication. This entire process that I'm going to explain to you is number 1, or formation, where we're actually forming the replication fork. If we were to draw them out, he would look something like this. Don't forget that DNA strands are anti-parallel, so we're going to have 5' to 3' meshed by 5' to 3' on the opposite strand. The fork is where my DNA parent strand splits into two template strands, and the enzyme responsible for carrying this out is going to be called helicase. Move on to step 2, or initiation. In initiation, what's actually going to happen is that we're going to lay down some starting blocks made out of mRNA using an enzyme called primase. And we need these starting blocks so that the next enzyme knows exactly what to do. What primase is going to do is on the 3' ends of his DNA strands, he's going to add in just a little bit of RNA. Something like this. He sees an A on the DNA template strand, he's going to add in a U because it's mRNA. He sees a T on the DNA template strand, he'll add in an A, and it'll be just enough nucleotides for our next enzyme to get to work. The same thing will happen on this second template strand here. I'll get an RNA primer, but remember it attaches on the 3' end of your anti-parallel strands, so that means that he's going to be connecting in over here. G will have a C, C will have a G. The second step is called initiation because we're finally starting the DNA replication process. Its main enzyme is going to be not polymerase, don't ignore my writing, RNA primase. He's going to attach to the 3' end of your template strands, and he's going to add a little bit of RNA called an RNA primer. Watch, I'm going to correct myself. This is RNA primase because he primes it and gets it ready to go. RNA primase is going to add a short RNA primer or a matching complementary RNA segment to the 3' end of your DNA template strands, and this is called initiation. the real fun of replicating DNA or making a copy of it is going to begin, and this is with an amazing enzyme called DNA polymerase. DNA polymerase always travels from the 3' to the 5' direction, 3' to 5'. He will attach onto the RNA primers, and then he will just start adding in complementary nucleotides. FYI, when I'm talking about how the base pairs match in DNA, A's with T's, or straight with straight, C's with G's, or curvy with curvy, I will often refer to them as being complementary base pairs. That's so nice of y'all to pay me some compliments. No, but seriously, in this case, when the base pairs are complementary, that means that they go together well. They fit together well. So A's with T's, C's with G's. Example, if he sees a C, he will add in a DNA G. I'm actually going to use blue so that we understand that it's DNA as well that's coming in. So on and so forth. Same thing over here. He's going to attach to his RNA primer here and move from the 3' to the 5' direction of the template strand. For a C, he'll get a G. For a G, he'll put in a C. For an A, remember he's making DNA, not RNA, so he's going to put a T. And for a T, he'll make an A. And then he's going to end off with a little 3' there. Just realize this might be helpful for you to know. Our template strand is our parent DNA. He's called a template because I'm using his code to build the new strands. So, this whole process is our third step of DNA replication, also known as elongation, because I'm actually building the DNA daughter strands. Our replication fork stays the same with 3' to 5' anti-parallel. Our main RNA primers are still posted at the 3' ends of the template strand. And then our main enzyme is going to be our DNA polymerase, who adds on complementary DNA bases in the 3' to 5' direction on the template strand. Now, you may notice that we run out of room here at the replication fork, and we need our good old friend helicase to come back and keep separating the DNA strands, the DNA strand into two template strands. So DNA helicase is going to go along his merry way, break some more hydrogen bonds between the complementary bases, and add them in. Now that helicase has opened up the replication fork even further, we might notice that an interesting problem occurs. On this strand, I will notice that DNA polymerase, he can just continue adding in his complementary DNA nucleotides from 3' to 5'. Right? E's with A's, so on, so forth. No problem. And he can wait until DNA or helicase opens up a little bit more of the strand, and he'll just keep going in this general direction. Yeah. On the other strand, though, we are actually traveling in the opposite direction because DNA is anti-parallel. So in order to go in the 3' to 5' direction that DNA polymerase usually travels, I'm going to have to actually go opposite. And since DNA polymerase always needs a DNA primer to latch onto, he's going to have to wait until helicase opens up the strand a little bit more in order to actually fit the next primer in there. And once that primer is in there, he'll be able to bind on and add in just a few more nucleotides. And this continues for a while with these two different strands. So if I was to allow helicase to do his job just a little bit further, continue separating the parent strand of DNA into two template strands. Once again, I would notice that this strand here, he can just continue going without any hesitation. Whereas this bottom strand here is going to have to wait for some primers to be laid down by primase. And then he's going to have to work from the 3' to 5' direction opposite of this guy. In this phenomenon, where we see DNA polymerase only moving from 3' to 5' in the template strand, he's going to have one smooth, continuous strand with no breaks, and then he's going to have one other strand that's going to be like primer, a little bit of DNA, primer, a little bit of DNA, primer, a little bit of DNA, all because he's traveling in the opposite direction, and he has to keep waiting for helicase to open up the rest of our replication fork. These two types of strands have very special names. So the one that is very long and continuous and can keep going straight on to 5' is called the leading strand because he leads straight ahead and he doesn't have to really wait for anyone. The other strand is called the lagging strand. Lagging is like that really annoying younger sibling who's always lagging behind and you always have to wait for them. That's what it means to lag. Or probably something more pertinent to you guys is a laggy computer, because we all are running Microsoft Teams and Pear Deck and the Hub at the same time. If you're waiting for things to load, if they're moving very slowly, it's lagging. Just like that strand that is waiting for helicase to open up the fork some more so that primase can put in another primer so then he can add on another short piece. Those little short pieces actually have very special names because they were discovered by a Japanese scientist. They're called Okazaki fragments. So, these two strands are called the lagging strand and the leading strand. The lagging strand replicates in small fragments called Okazaki fragments, and that is because he's waiting for helicase to open up the replication fork. The leading strand replicates continuously in one single take from 3' to 5'. Our last step of termination. We've created the complementary DNA strands, one leading, one lagging behind. There's only one thing that's making it not quite right, and that is the presence of mRNA still within our DNA strand. That's a big no-no. To help us repair that problem, we're going to have two other enzymes. One is an exonuclease, represented by an eraser. The second is a ligase, represented by tape. Exonuclease, exiting nuclease, the nucleotides, is going to basically remove your RNA primers. I know, I was so clever. I wrote with pen and then decided to use an eraser as an analogy. Let's go find some whiteout. Whiteout procured. You have been demoted from being an exonuclease. So, like I was saying, this is an enzyme. He doesn't even want to be an exonuclease. This is an enzyme that's going to remove RNA primers so that they are actually taken out of the DNA template strand. Same thing over here on the lagging strand. You'll have a lot more work to do because there's a lot more primers. Okay. And this allows DNA polymerase to come back in and kind of fill in that gap where there used to be RNA. And it works this time because he has some nucleotide bases to attach to already. The final step comes in with ligase. So if you notice that you've went in, you've made some DNA strands, and then you've taken out RNA and you've replaced it, there's actually going to be little gaps between these guys here. And they just need to be sealed off with a little bit of help from ligase. He's a bit like tape. He's going to just make sure that all of the new nucleotides get put together correctly. Ligase and exonuclease also act like proofreading tools. They kind of make sure that there's no errors in the matches between the complementary bases for our new strands. All right, fourth and final step, also known as termination because we are ending. Along our replication fork, we need to get rid of the mRNA primers that were left there by primase. The enzyme exonuclease will do that for you. This leaves some gaps in the DNA, and so those will be repaired by an enzyme called ligase. I'm going to do some final touches. We got one last little primer where exonuclease needs to excise him, take him out. So you might notice that when we take out the very last primer for this lagging strand, it's over here on a 3' end and there's nothing for DNA polymerase to attach to, which means every time you replicate your DNA, you actually get rid or lose some of the nucleotides there. Sounds absolutely horrendous because cells divide sometimes hourly. And if that means you're replicating your DNA hourly, then you're losing nucleotides hourly. Yikes. Now, fortunately, the cell has a safeguard, and it's something called a telomere. Basically, at the 3' end of every DNA strand, there's just a long mumble-jumble of nucleotides that don't even code for a protein. They're just there so that every time you replicate, you can lose a few, and nothing will happen. At least nothing in terms of you're not losing actual genes that code for actual proteins, you're just losing random nucleotides. Now, as you can imagine, as a person gets older and older, they might run out of telomeres, which means they might actually end up starting to lose some of those vital nucleotides for genes as they get older. And this is some theories, and some scientists enjoy looking at this as to why people actually change or their bodies don't function as well when they get older. The idea is that they may have lost all of their telomeres, and now every time their DNA replicates, they're actually losing nucleotides for vital genes. If you're interested in looking into things like aging or Alzheimer's or Parkinson's, a lot of research has been done on telomeres, so go check it out. And there you have it. Definitely not quite as neat as just copying a pair of cards, but I hope it actually kind of made sense how you can make a copy of your DNA from one single strand. We'll see this again in class in a little bit together instead of on a video. And then also, I want you guys to practice all of these with your DNA replication assignment on the hub. Good luck. Hit me up in the chat if you've got questions. Just FYI, I swear my knitting bag is usually not that messy. I mean, just take a look at my Sewing Notions box. I stay organized. I try to, friends. Fortunately for you, your DNA stays more organized, perhaps, than Miss Lim, in an effort to avoid mutations. That zipper was bothering me, as is this lace. Now we're better.